A lovely and warm good morning to our most valued and worthy dignitaries, management committee members, sponsor AICTE, respected participants, principals from all over India, and my beloved colleagues. I, Rajaram Das, Assistant Professor RIPS, on behalf of entire organizing committee, welcome you back to the ninth day of Series 3 online FDP sponsored by AICTE and organized by Roland Institute of Pharmaceutical Sciences. To mark this event a traditional beginning, let us begin with a virtual presentation of lamp lightning, followed by Ganesh Vandana.
शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तुते दीपज्योतिर्नमोऽस्तुते शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः
वक्रतुण्डमहाकाय सूर्यकोटिसमप्रभा निर्विघ्नं कुरु मे देव सर्वकार्येषु सर्वदा सर्वकार्येषु सर्वदा सर्वकार्येषु सर्वदा
With the blessings of Lord Ganesha, let us start today's program. Dear participants, today we have our first eminent resource person with us, who is a very well-known facet in our pharmacy fraternity, and such a person does not require any kind of formal introduction. Before inviting him to share his valuable talk, it is my privilege to introduce today's resource person, none other than Professor Dr. M.E. Vanoji Rao sir, to all of us. Dr. M.E. Vanoji Rao sir is graduated in pharmacy from Andhra University in the year 1980 and his post-graduation from Andhra University in the year 1983 with first class and distinction. Following a brief tenure of 2 years in the industry, he switched over to teaching profession in 1985. After working as a lecturer for 1 year in College of Pharmaceutical Sciences, Mahuda, he joined Roland Institute of Pharmaceutical Sciences, Berhampur, as the founder principal. Sir has obtained his PhD from Kakatiya University in the year 1997, and soon after he was promoted as professor. Sir continued in the same institute as professor cum principal till 2019. Dr. Rao has guided more than 65 M.Pharm dissertations and 18 PhD scholars. He has published about 80 research papers in various journals of national and international repute. He is PhD adjudicator and PG examiner for different universities, including Berhampur University, Biju Patnaik University of Technology, Jadavpur University, Andhra University, Kakatiya University, Rajiv Gandhi University, JNTU University, and Geetam, etc. Sir has completed 5 projects worth 40 lakhs sanctioned by AICTE in New Delhi and successfully organized 4 members of 2-week staff development programs along with numerous workshops and conferences in this institute. Sir is instrumental in starting AICTE approved postgraduate program, that is M-Pharmacy course in the state of Odisha for the first time during the session 2000 and 2001, and it started in Roland Institute of Pharmaceutical Sciences, Berhampur. Professor Rao has served as chairman Board of Studies, Berhampur University, member Pharmacy Council of India, member Research Committee, Berhampur University, member Expert Committee, UGC, chaired scientific session, Indian Pharmaceutical Congress in several occasions. Presently, he is continuing as member BOS Academic Council and Dean and Faculty of Pharmacy, Biju Patnaik University of Technology. Professor M. E. Bhanoji Rao sir has 35 years of professional experience, including 27 years of teaching and 21 years of intense research. He is a life member of professional bodies such as Indian Pharmaceutical Association, Association of Pharmaceutical Teachers of India, and Indian Hospital Pharmacist Association. At present, Professor Rao is working as Principal and Professor, Calcutta Institute of Pharmaceutical Technology and Allied Health Sciences, Howrah, West Bengal. With this brief introduction, may I now request Professor M. E. Bhanoji Rao sir to kindly join us on board to deliver his valuable talk.
Thank you. Thank you, Mr. Das, for your nice introduction. Yes, I am very happy to associate myself today as a resource person for this program. And at the outset, I would like to thank Professor Ravi Kumar, the coordinator, and the co-coordinator, Dr. Mohan, and the faculty members of Roland Institute of Pharmaceutical Sciences. And I also welcome all the delegates to this small talk, which whatever information I have uh, uh, on this particular topic of gastrointestinal drug delivery systems to share with you. So I am very much thankful to organization once again. I have a long association with the Roland Institute of Pharmaceutical Sciences from the inception, and almost 32 years I spent. I had my professional career in Roland only. Therefore, the bond is very strong with that institution, whether it is students, or non-teaching staff, teachers, or management. So once again, thank Ravi Kumar and the colleagues for inviting me to this program. Yes, I would start this program, my talk. So I request, I think whenever this slides to be operated, please cooperate with me. Yeah. Once again, good morning to all of you. So gastrointestinal related drug delivery system and overview and advances. Yeah. So about the introduction, as you know, oral delivery remains the most preferred and convenient route of drug administration. Oral route of many advantages from patient point of view, physician point of view, as well as manufacturer. If you see the patient's preference, oral dosage forms are very easy for administration, convenient for carrying, that is where very much portable, and cost effective, that means they can affordable when compared to any other dosage form. Oral dosage forms are, particularly solid dosage forms, are very much affordable to the patient. When we refer to physicians prefer because it is particularly non-invasive, and there is a flexibility of dosage. you can change the doses as per the requirement of the right, and therefore the high patient complaints which the physicians uh, want, that is fulfilled. So this is the preference of physicians. Coming to the manufacturer's viewpoint of view, there is a lot of flexibility in formulation, feasibility of high productivity. Then there are less rigid controls and less stringent regulatory requirement. Of course, the oral route is the most convenient, but it is not so simple route. There are number of barriers of the gas tract which are difficult to overcome. So let us discuss a few of these difficulties, barriers. First of all, the GI tract has having varying pH from starting from pH to acidic pH to the smallest one, which is gradually increases to alkaline side. There are presence of different enzymes in the stomach, small intestine. Then there are physical barriers of epithelium. Then weighing surface area is also changes, and particularly small upper small intestine is the area where large surface area where the absorption is mainly takes place. Apart from this, there another important factor is that pre-systemic or the first pass measurement. So these are the various limitations, or you can say, because the pH conditions and enzymes, these are very harsh conditions which the drug has to encounter before it is getting absorbed. And also there are various physical barriers, and as I told you, many drugs are not stable in these harsh conditions, and also the bacterial flora that is in the present in the gut, as well as the liver, which the drug has to undergo first pass metabolism. So these are the various factors which can limit this oral drug delivery system. Of course, the advantages are overweigh the disadvantages. Therefore, this it is the most widely accepted route for patients, physicians, as well as the manufacturer. So, what are the gastrointestinal? What is the problem? Why they are very so promising, and they are having the potential to address many of these oral delivery needs through this by gastric retaining of the drug delivery systems of the devices. So gastric to retentive drug delivery system facilitates prolonged and continuous and complete absorption phase of the drug in the upper parts of the GI tract. Then gastric retention avoids significant heterogeneity throughout the GI tract, including the pH, bacterial flora, GI transit time, enzymatic activity, and surface area, all of which can influence absorption. Another very important thing is improving the biopharmacokinetics of the drugs, particularly with a narrow absorption window or narrow absorption therapeutic index, and achieving efficient oral delivery for poorly soluble drugs, which can lead to significant improvement in the efficacy and safety of the treatment. So, as it has been recognized that many drugs have the absorption window, and that means in a particular part of the small intestine, particularly in the proximal intestine, these drugs have getting absorption, and other in other part of they are to a very small extent they are. Therefore, they possess this particular behavior, and therefore it is necessary that the drug delivery system remains in the stomach and release the drug before they pass this absorption window. Similarly, in case of drugs having narrow therapeutic index and window, that means the safety margin between the efficacy and the toxic level. If that is very narrow, then easily they can cause drug toxicity. Therefore, when these drug delivery systems, because they release the drug in a continuous and in a steady manner, therefore they can be a steady state can be maintained within this therapeutic range, so that this problem of either underdosing or the overdosing can be eliminated. On the third, in case of narrow, poorly soluble drugs, it can lead to significant improvement in the efficacy and the safety of the treatment, because the drug remains in for a longer time, though it has inherent poor solubility. It has it receives longer time for getting solubilized, and therefore they can be absorbed effectively. So these are, and apart from this, uh, drugs which require which act in the locally, they can be effectively formulated in forms of drug related this active gastro-related drug delivery system, because particularly uh, in case of H2 inhibitor, that means drugs which acting against anti-ulcer drugs, and drugs antibiotics which act against this H. pylori. Then coming to the various nomenclature in case of controlled drug delivery systems, we have come a long way from conventional drug delivery to sustained release, controlled release, and there are so many synonyms: sustained release, extended release. So therefore, USP definition makes it very clear that what are these systems are, what are the different between among themselves. So as per USP, the modified release dosage forms are defined as those whose drug release characteristics of time core and or the location are chosen to accomplish therapeutic objectives which are not offered by the conventional dosage form. So it is a very broad definition. That means any dosage form which is other than the conventional doses, where there are the requirements for drug control release and the release at the particular location. Such type of dosage forms to accomplish the therapeutic objectives, the modified release drug delivery systems are designed, and how this USP defines the systems. Whereas the another very commonly used term, that is extended release. These are the dosage form, those which allow a twofold reduction in dosing frequency or increase in patient compliance or therapeutic performance. So it means that if the drug is having a conventional dosage form, the dosage frequency of 4 times a day, if it is reduced to 2 times a day, it can be called as a leg. Similarly, the patient's compliance, if this is doubled or the therapeutic performance is increased, such a dosage form can be defined as extended release. The other terms like control release, prolonged release and sustained release, or these are interchangeably used with extended release. So the most, scientific way of expressing is the extended release, and others they are come under modified release, drug delivery system. So the gastroretentive drug delivery system is is a modified release as well as extended release. Now coming to uh, any development of a formulation for oral control release, one must have the knowledge of this biopharmaceutical classification system, which is very important a tool to judge and decide what type of dosage form and what are the candidates for selected can be for this either extended release or modified release, whatever the control release that is being formulated. So let us discuss about what is BCS. The active solubility and the intestinal permeability of drug substances are paramount important. This is as suggested by Amidan, and accordingly, the drugs are classified into four categories depending upon the solubility and the permeability extent. The class one drugs belong to high solubility and high permeability. That means these drugs are very easy for formulation. As a pharmacist, we do not encounter any problem or issues regarding the formulation of. Hello. Yes. Uh, so these are the best cases. They come under best case because they have good form solubility and good permeability. So they do not possess any or pose any formulation problems. Now coming to class 2, these drugs have low solubility and high permeability. And many of these, the currently available drugs are around 40 to 50%. They belong to this category. Many of them have this problem of low solubility, but they are having good high permeability. So the problem here is to solve the solubility problem. Then it can be successfully formulated as a control regulator. Class 3 drugs, they have good solubility or a high solubility, and but having low permeability. Therefore, the problem is to address the permeability or to increase the permeability. So nowadays we have a set of both chemical and physical permeation enhancer. So if those are rationally really utilized, then the permeability can be enhanced and the formulation can be successfully done. The class 4 is the, I think, having both this problem of solubility as well as the permeability. So therefore, this is the worst case class. You have to solve both the problems of solubility and permeability. So the available techniques of improving solubility and enhancing permeability have to be utilized in order to formulate drugs of this category. So just understanding the classes, I think it is not sufficient. We have to know what is the high solubility and what is low solubility, high solubility, or what is these things. Let us focus on this. High solubility means solubility in the pH range of 1 to 8, and the dose, which is, the complete dose should dissolve into 250 ml of, in the medium, in aqueous medium, in all the pH, then that may be called as a high soluble drug. Then coming to high permeable, here if the permeability coefficient P is around 4 to the 4 into 10 to the power of minus minimum mm per second, can have absorption up to 90% of the dose. So such drugs may be called as highly permeable. Similarly, low aqueous solubility, that means those having usually less than 1 mg per ml solubility in aqueous solubility, they can be defined as low soluble drug. And low permeability, that means low permeability efficient coefficient, which is less than zero point 0.5 into 10 to the power of minus 6 millimeters per second, is expected to have around 20% of absorption only. So that is low permeability. So accordingly, we can define these four classes and understand their solubility and permeability properties. And these have been actually defined by studies on cell line cultures like Caco2 tissue culture model. Basing on that, these have been formulated to understand what is a high solubility, high permeability, or low solubility and low permeability. So these are very important studies which have to be done during the pre-formulation to understand about that the drug absorption of the drug. That is very important. So apart from the solubility and permeability, what are the other criteria that is also to be considered? The biological half-life, 4 to 6 hours. Those having T half 4 to 6 hours, they can be a good candidate. Similarly, if the dose is not exceeding 325 mg per, in order to restrict the size of the delivery system, because the delivery system or the tablet or the must be convenient for swallowing. Therefore, the dose, the upper limit of any controlled release can be 325. It is the optimum. Maybe having slightly higher, that is not, does not matter, but this is the optimum level. Similarly, no first pass metabolism. So those drugs which are having low or very less permeable first pass metabolism, they are good candidates. Similarly, the stability should be very good in, because as earlier discussed, the pH, which changes in the GI tract, and the enzymes which are present, and the bacterial flora, all these have influence on the stability of the drug. If the drug is stable in all these conditions, that's a good candidate for controlling. Similarly, we have this. What are the optimal absorption criteria? Let us discuss these things also. The first one is high permeability coefficient. It's a one very important criteria for absorption. Passive diffusion or direct transfer, or known carrier-mediated transport mechanisms. So before formulating, we had to have these studies. What is the mechanism of drug absorption? Whether it is passive diffusion or carrier-mediated transfer, that is active system, like that. Similarly, high solubility in aqueous media and over a wide pH range of 1 to 7. This is a very important criteria. Similarly, as I earlier told, no or low degradation or metabolism in the intestinal lumen fluids, and low first phosphorus metabolism. Then complete absorption in the GI tract, which has to be studied under various with various animal models. In vivo animal models studies have to be conducted to know to what is the extent of the absorption. So if they are completely absorbed, then it is a good criterion for optimal absorption. Uh, we have many physicochemical properties which usually studied in the pre-formulation testing to know how best it can be formulated in a in a particular dosage form of our objective. So aqueous solubility, aqueous stability, extent of ionization, pK, pH, because most of the drugs API are either weak acids or weak base. Therefore, we have to know their ionization behavior and at various pHs, what is the pK. And then similarly, we have another physical factor, that is log P or partition coefficient, which gives an idea about this permeability, because all the membranes are lipoidal in nature. If the drug is in unionized form, they are expected to be better absorbed. So therefore, partition coefficient, it has a lot of relevance for permeability, which is often on water system. We have to determine the partition coefficient or the log P. Similarly, salt form, it is also polymorphism. What is the crystal form, whether it is crystalline or amorphous. So the stability and the solubility of the salt form, these are also very important for formulating a drug into a controlled process. So as we had to understand the pH conditions under which the drug has to subject it to after oral administration. So as we know, stomach, it is highly acidic conditions, and which usually in the fasting conditions it varies from 1 to 2, and in fed condition 2 to 5, depending upon the food material or the volume, pH of the food material, the pH, particle size, all these have influence on the pH. Therefore, in fed condition, usually it is expected that the pH varies from 2 to 5 in the stomach, and in upper small intestine, it ranges from 5.5 to 6.5, and in lower small intestine, it is 6.5 to 8, and in colon or the large intestine, it is 5.5 to 7. So coming to the definition of gastrointestinal drug delivery system. So gastrointestinal drug delivery systems are those which prolong the residence time of the systems to achieve complete drug release in the gastrointestinal tract. That means both stomach and small intestine, as well as to modulating the drug release rate as predicted by the system in order to obtain an ideal oral control release. So therefore, there are two things. One is the system has to be retained in the stomach, and it has to release the drug in a controlled and a continuous manner, so that the drug can be absorbed completely in the particular area or the therapeutic window, such that we will get the predicted drug release as per an ideal oral control release. So, what are the major advantages of these systems? The first one is reduced variability of the drug release. So, drug release will be steady in a controlled and continuous manner, and there will be no much fluctuations in the levels of the drug for a over period of long time. Similarly, local drug delivery in the stomach and in the upper or proximal GI tract. So drugs which have to act in the particular local place of stomach or small intestine. Similarly, this can be a good candidate for, and that is an advantage for gastrointestinal system. There is enhanced absorption and bioavailability of having restricted or narrow absorption in the GI tract. So this is a very important advantage of these systems, because as I already told, once the drug passes its absorption area, there is no scope for the drug to be getting absorbed, because in the distal small intestine or in the large intestine, the absorption is to a very low extent. Therefore, the drugs having narrow absorption window are the best suitable for this type of system. And there is improved absorption and bioavailability of poorly soluble. As I told you, because the drug has the longer time in the stomach before it is passes the small intestine. Though it's inherent poorly soluble, the chances of getting more drug getting solubilized is possible. Therefore, such type of drugs can be formulated for improving their bioavailability as well as absorption. So let us see what happens when a gastrointestinal system is administered. The as related to drug release prolong the gastric retention or residence time of the system to increase contact time for the drugs which act locally, such as gastric acid secretion inhibitors, the anti-ulcer drugs, and etc. To increase absorption for drugs that have narrow absorption window, for example, chlorothiazide, riboflavin. There are some number of examples are there. I can show you those examples at a later stage. For better absorption, drugs having low solubility, which are in the intestinal fluid, that is already been discussed. Now, what are the physiological factors to be considered? That is pH and surface area available for absorption, and very importantly, the gastric motility pattern, which depends upon phosphate and fed conditions, and in further it depends upon the nature, quantity of food material, food particle size, etc. So gastric emptying time for a regular meal is 2 to 6 hours, but it varies from person to person. Individual variation is a lot of variation is there. And another physiological factor is digestive migrating motor complex IMCCs. This is a phase. I mean, in fast condition, there is stomach is under dynamic condition. It undergoes 4 phases under IMC. Phase 1 usually is an inactive phase where there is no much movements at all, which lasts for about 42 to 60 minutes. And the phase 2, there are some intermittent and a few contractions, and this phase lasts for about 30 to 45 minutes. And the third phase is the most important phase, where there is a sudden increase in the gastric motility, and with a high, emptying capacity, uh, and it is usually called as a, uh, I mean, they call housekeeping space. That means it removes whatever the indigested food materials there, that is removed slowly by this intense movement of the gastric, stomach movement, and it lasts for about 10 to 15. And this is the most important phase, where these gastrointestinal duct delivery systems have to withstand or survive this onslaught. So once these movements have been withstood, the delivery system can remain in the stomach for a longer time. And the phase 4, it is an intermediate phase within the fourth and the first phase. So the entire 4 phases of IMMC, let me say, interdigestive mitotic motor complex system, lasts for about 120 minutes. And this is a very important physiological factor for the gastrointestinal delivery systems. So therefore, now what are the various approaches for these systems? Let us start with the intragastric floating systems or hydrodynamically balanced systems. Then mucoadhesive system, high density systems, magnetic systems, unfolding extendable or sellable systems or expandable system, and superporous hydrogel system. Apart from this, there are other approaches like having different shapes, etc. Those have practical difficulty. Even in these cases, these high density systems are practically difficult to formulate. And magnetic systems are not very convenient for the patients, because you have to externally apply a magnetic field to keep the system in the stomach, and therefore this is practically, or it is not a feasible system. And similarly, if you see the intragastric floating system, there are a lot of advancements. Therefore, they are working well. Whereas this hydrodynamically balanced system, that means they are having less than one density, they can float in the gastric contents. And of course, single unit HBS has not much application, because single unit systems, they have the drawback, all or none. That means if it may be released, or it may not be the drug may not be released, because it is a single unit, and there is a lot of variation in gastric emptying from person to person. Therefore, it is very difficult to get reproducible or predictable drug release. There are advances in gastric floating system. That means floating system or buoyant systems. There are a number of systems, again gas or non-gas systems, effervescents, etc. those things I think we have already been well covered. I mean, in various textbooks. So I'm not going to deal those things in detail, but only a few advances. Similarly, mucoadhesive systems. Here we have certain advances which I will discuss in at a later stage. High density system, that means those having more than 1.3 grams per cubic centimeter. They are high density systems and they can settle in the stomach. But as I told you, that's practically very difficult. Similarly, magnetic systems also having lot of limitation. Coming to unfolding, extendable and swellable system, they have got a lot of sexes, and this can be discussed in our later stage. Similarly, superporous hydrogel system. These are also uh, an advanced system which can be discussed in our this presentation. So, what are the physico-chemical and physiological factors that have influence on the viability of the drugs after oral administration? So this is I mean a total information about these two factors. Usually we studied physicochemical properties of the API, like which are very important parameters like solubility, particle size, hydrophobicity, molecular size, pK, chemical stability, crystal or salt form, complexation. All these physicochemical factors have to be studied before we are embarking on this formulation. Similarly, the physiological factors are equally important and to understand so that how the drug delivery system behaves after oral administration. So therefore, what is the surface area available for absorption, transit time and motility, because there is an important transit time, right, is the most important criterion for or retentive drug delivery system. We have to enhance the transit time. Then only we can expect the drug to be released completely. Then intestinal secretion, as I already told, there are enzyme and pH conditions, and these are very harsh conditions which can influence the drug on the stability. Similarly, enzymes, even bacterial enzymes, they can degrade the drug. Similarly, membrane permeability. What is the mechanism of drug absorption? These have to be understood properly. Similarly, food and food composition, which has a lot of influence on the emptying rate, gastric emptying rate. Therefore, it has to be studied properly. And what condition, whether it is fasting or fed condition. And, and in both the conditions, the system has to remain in the stomach for a longer time. Similarly, bacterial flora and liver uptake, that is nothing but first class metabolism, to what extent the drug undergoes. So these are the physicochemical factors as well as the physiological factor, which has to be understood properly and studied before we are embarking on the assignment of formulating a controlled drug delivery system, including gastroretentive drug delivery system. So, for achieving the efficient oral delivery system, oral formulation, there are certain challenges in the drug development. So, as we know already, drugs with narrow absorption window, drugs having narrow therapeutic index or window, and drugs with poor aqueous solubility. These are the very important challenges which have to be addressed uh, in order to successfully formulate or design gastrate into drug delivery system. Now, there are certain examples of this narrow absorption window drugs. So, we have furosemide, which is mainly it is absorbed in the stomach. Riboflavin, ripaglamide, metformin, gabapentin, ciprofloxacin, ofloxacin, levodopa, and aceclor. So various locations in the GI tract where they have their absorption window. These are a few examples I have given, but there are a number of examples are there in the literature. These are the drugs having narrow therapeutic index. As I told you, the margin difference in the efficacy and the toxicity, safety and efficacy is very narrow. So therefore, these are the drugs which having that problem and which should be properly addressed. And glibenclamide, ibuprofen, ketoprofen, cyclophenol, carvedilol, ketoconazole, naproxen, rifampicin, etc. I'm sorry. These are the sorry. Those are the BCS2 drugs. BCS2 drugs having low soluble, which just now I have given the example. And these digoxin, digitoxin, aminoglycoside antibiotics, rifampicin, carbamazepine, phenytoin, lithium, warfarin. These are all having narrow therapeutic index. So therefore, they must be very carefully designed. And in clinical pharmacy, we have therapeutic drug monitoring. So these drugs require therapeutic drug monitoring. Constantly, their drug levels have to be monitored to ensure that they are within the safety margin. So therefore, gastroretentive drug delivery system for the drugs can address the problem of narrow therapeutic window of these such drugs. Actually, once again, yeah. Yeah, here I am going to discuss some of the systems which are advanced systems or which are I think should be discussed, because other things you already know very much very well, and therefore only some advances and some in important advancements only I am going to discuss. These microballoons, which, as you know, these are very small porous systems, hollow microspheres, which can float. So this is the one type of floating system, and they have a lot of promise and potential for, as control release by this gastroretentive system. So the preparation, just now I have outlined it. How to prepare this? I will just briefly discuss. So the drug is to be dissolved in an organic system. In this particular preparation, ethanol and dichloromethyl taken in 1:1 ratio, and the drug is dissolved at the room temperature, as well as the polymer utazide S100. here is taken as the polymer for preparing the microsphere, and it is mixed with HPMC in 9:1 ratio. Here HPMC to control the drug release. So utrazide forms the microspheres, whereas HPMC is mixed to control the drug release. So both these polymers taken in 9:1 ratio, along with the drug, they dissolved in ethanol and dichloromethane, taken in 1:1 ratio in at room temperature. You'll get the drug polymer solution. This is added to aqueous polyvinyl alcohol. It is usually less than 1% solution, and then we will form a solid droplet. Simultaneously, we have to agitate, and continuous agitation subject to agitation as well as at 40 degrees centigrade to evaporate the organic solid, particularly dichloromethane. Once these solid droplets which are formed are subjected to evaporation, dichloromethane, when evaporated, forms certain pores on the this microsphere, and on further drying, the water is also removed gradually, and therefore the particles become hollow as the solvents escape, both organic and aqueous water. When they escape, we'll get the hollow microspheres that is known as microbillion. So these microbillions um, uh, reportedly have very good floating properties and remain in the stomach for a longer time. So this is a patent technology. Hello. Yeah. So this is briefly how can we prepare microballoon. So drug is incorporated along with the polymers in the organic solvent, subjected to evaporation, and before that it is added to aqueous polyvinyl alcohol solution. So emulsion evaporation technique is. And finally, when the water is removed, the microspheres, hollow microspheres are formed, which are nothing but hollow microspheres, which are having good floating property. And here HPMC is mainly used to control the release of the drug. So this is a promising approach for floating system for gas storage into drug delivery. Next, I am going to discuss about mucoadhesive liposome. As I told earlier, compared to single unit system, multiple particulate systems have a number of advantages because these particles are dispersed in the stomach, and they are emptied in a random fashion. Therefore, they can remain in the stomach and can release the drug in a continuous manner. Unlike single unit, as I told, all or none, it is not the case. Here, the microparticles are distributed in the stomach, and randomly they are emptied from the stomach. So during the drug, particles remain in the for a longer time, and they release the drug for an extended period before they cross the absorption site. So let us see these micro particles systems, multiparticulate systems. Mainly they are microspheres, nanosphere, and liposomes. Here I am going to discuss about nucleoadhesive liposome. Just I will give the outline of this, how these liposomes are prepared. So this the very conventional method of liposome preparation, that is thin film hydration method. So the organic solvent phase consists in this we have to dissolve phospholipids like L alpha palmitoyl phosphatidylcholine DPCPC and dicetyl phosphate. Hello. Yeah. Yeah. Organic phase is the organic phase. Any organic solvents like usually ether, dichloromether, alcohol. These are to be selected, and the phospholipid and the polymer, they are dissolved in the organic. And the organic solution of this phosphate lipids taken in a rotary evaporator and subjected to evaporation continuously to form a thin film of the lipid on the surface of this the vessel. Once it is completely dry, that the solvent is removed, a thin film of the lipids is formed on the vessel. The aqueous phase consists of the drug in a suitable buffer like phosphate buffers, so pH 7.4. This aqueous phase is added um, all at a time, okay, to this thin layer of lipids, and with continuous stirring, it will form the liposomes. And these liposomes are heterogeneous in their size. That means they vary in the size. Therefore, it is necessary to homogenize the liposome, this heterogeneous liposome, and they are subject to homogenization or sonification, and which result in the uniform distribution of the size and formation of liposome. And finally, coating has to be done with chitosin, which is an mucoadhesive polymer. And therefore, once these liposomes are coated with chitosan, we will get the mucoadhesive liposome. That means liposomes coated with a mucoadhesive polymer like chitosan, which which can adhere to the mucus of the small stomachs and remain in the stomach for a longer time. So this is a micro mucoadhesive system. Apart from liposomes, we can have microsphere, nanosphere, using polymers such as sodium CMC, sodium alginate, carrageenan, and carbopol 9334P. These are some important bioadhesive polymers. So we can pick a microsphere, so nanosphere, liposomes, and finally coat them with bioadhesive polymer so that we will get bioadhesive systems for gastroretentive system. Uh, Next, I will discuss another approach, which is but superporous hydrogel. As the word says, superporous, that means as soon as they are admitted, immediately or instant and hello, yes, this preparation of superporous hydrogel. This, as I told you, this super porous hydrogel have the property to swell instantaneously when administered into the stomach, and they swell to a larger size than the sphincter. The human sphincter diameter is about 2 centimeters. So larger than 2 centimeters, they will expand, and they are obstructed from entering in the small intestine, and thereby they remain in the stomach for a longer time. So let us see how these superporous hydrogels are prepared. It is nothing but a copolymer having the property of gelling or expanding instantaneously to a large extent. So let us see what are the monomers required for the preparation of this copolymer. The one monomer is acrylamide, and the another monomer is sulfopropyl acrylamide potassium salt, SPAC, SPAK, SPAC. Sulfopropyl acrylamide potassium salt. This is one polymer, monomer, and acrylamide is another monomer. These are to be cross-linked to form a copolymer, which possesses the property of superporous gelling. So what are the requirements? Let us see. Apart from these monomers, there should be it should be in an acidic medium. The polymerization takes place. Therefore we have to add acid, linking agent, foaming agent, and the initiator to start the reaction initiate. So therefore, once they are taken, the polymerization starts, and it's accelerated by increasing the pH by adding sodium bicarbonate. So it is a very critical phase for formation of this copolymer. Sodium bicarbonate has to be added at the right time to accelerate the polymerization process with the increase in the pH. And once the reaction stops, superporous gels are formed. It is nothing but a copolymer known as polyacrylamide SPAC. Yes. So this copolymer is having the superporous gel property, and this superporous gel is further coated with 10% mucin, because these superporous hydrogels should have a slippery or slimy coat, so that even the stomach contracts, it will not slip into the small stomach. So there the surface is made slippery by coating with 10% mucin at 130 degrees centigrade. And it should also have mechanical strength to withstand the IMMC, which I have discussed earlier. So therefore, this contractile motility of the stomach. Therefore, by adding Agdisol in the reaction mixer, it gives strength, mechanical strength to this micro superporous gels. So it's mechanically strong, becomes mechanically strong, as well as by mucin coating, it has a slippery coating, and which helps in these retaining in the stomach for a longer time. And once these gels are formed, they are dried and filled in a 000 capsule size, so that they can be conveniently administered. So once they enter the stomach, immediately they swell and retained in the stomach without escaping into small intestine due to their larger size than the sphincter. So these are the few advances which I wanted to discuss. Apart from the another recent development, which is a patent technology. Hello, yeah. Um, this is known as AP technology, or accordion field technology. It's a recently developed technology in Israel, and it has been patented, having a lot of promise, already undergone some clinical trials, and proved to have gastric retention up to 12 hours with controlled release of the adulterate. So just briefly, I will discuss about this accordion pill or AP technology, platform technology. Hello, yeah. This is a platform technology, that means is any drug satisfying the selection criteria can be loaded into the system, and it will perform as a gastroreticulated delivery system. It is basically a multi-layer structure with a convenient shape and styles, having record mechanical property. It is a number of layers it has. Inner layers consisting of drug-loaded layers, and the outer layer is for opening the system, and also it contains drugs for immediate release. And why they have named it as accordion pill is, I think many of you might be knowing, accordion is a instrument, musical instrument, having different wave-like structure, continuous wave-like structure, which can be folded and expanded. You can see in the literature what is an accordion. So because of that, the resemblance, like wave-like structure, a continuous wave, which is also known as undulating structure. Undulating means a film having number of folding, which can be squeezed as well as expanded. So that system has been developed and patented as accordion pill. It is a unique film-based gastroretentive oral drug delivery system. As I told you, these layers are made up of biodegradable polymer and folded into an undulated shape and is filled in a capsule. Finally, it is available in the form of capsule, and this capsule, when administered into the stomach, the capsule will dissolve, and the system it opens or unfolds, and it expands, and therefore it cannot escape into the small intestine because the size is bigger than the sphincter, and the outer layers immediately release the drug for which is available immediately. Um, then subsequently, the inner layers, they unfold, and the drug is released in a controlled manner and then continuous manner. And once the drug is completely released or emptied from the system, because it is a biodegradable system, it gradually dissolves and degrades, and it escapes from the stomach, and finally it degrades completely in the small intestine because the inner skeleton is made of enteric polymer. So in the small intestine pH, finally the backbone of the system also dissolves and completely vanishes. So before that, the entire drug is released in a continuous, in a controlled manner, and it is been claimed that hello, yeah. So this system is a patented technology and claimed to have lot of potential as a gastroretentive drug delivery system. So once again, just I will give a small um, detail about this system. So accordion pill is a novel gastric retention oral drug delivery system based on folded multi-layered layer. The phase 2 clinical trials in healthy volunteers have evaluated gastric retention and pharmacokinetic properties for efficacy and safety of two drugs, that is carbidopa and levodopa, in patients with Parkinson's disease have been evaluated. The system remained in the stomach for approximately 8 hours without any special meal requirement, and it has demonstrated that there is an improved absorption of levodopa compared to the immediate release dose system. So accordion pill delivery system is a promising and potential novel gastroretentive drug delivery system to improve pharmacokinetics and efficacy of drugs, particularly for narrow absorption window drugs, poorly soluble drugs, and the drugs having narrow therapeutic index. So I think that there is a small video clip. So this is how it looks like. You can see the different layers, the inner layers and the outer layer, and how it unfolds. And then I mean, it is actually filled in a capsule, and when administered, it unfolds, expands, and then releases the drug. It is retained in the stomach and releases the drug. I think this video clip, I think, can be. I think I request you to display that one, operate that video clip. Dr. Vismohan?
Running now, sir. Running, sir.
Huh? Can you
Yes, sir. It is running now. Yes, sir.
Okay. Because I am not able to see.
Yes, sir.
Okay.
Yes, sir. It is running, sir. No problem.
Yeah. Yes, you can see how it is. Yeah, once it is administered, it enters in the stomach. Yeah, the capsule has dissolved and the system has come out. So the drug is released from the layer. As you see, it is being blocked at the sphincter, but the drug is continuously being released. And finally, it escapes from the stomach and in the small intestine. It is completely dissolved and eliminated. So this is the technology, AP technology, which I found will be interesting. So with this, I have completed my presentation. But I would like to add that I have not covered the characterization or the evaluation part, because these are various different systems and having specific evaluation tests. However, we have to have the tests like drug content, the drug loading efficiency, and to locate that the system, whether it is there in the stomach or not. We have this X-ray graphics, photographics, and gamma scintillations systems, by which we can locate where exactly the systems are, whether they are present in the stomach or not. And apart from that, the most important release studies, in vitro release study, as well as in vivo studies have to be performed in various models, animal models. And finally, to know that its bioavailability, it can be done in human volunteers. So these are the important evaluation parameters. Apart from that, depending upon whether it is a flow test system or bioisodesis system or microparticle or nanosphere or liposome, accordingly those particular tests have to be performed in order to evaluate the systems or the characterization. So this is what I wanted to share with you, whatever small things I have acquired. Once again, I thank. Before that, a small conclusion, that is, advances in the design and formulation of gastroretinal drug delivery systems have demonstrated to be very promising to enhance absorption, bioavailability, and pharmacological activity of drugs having a narrow absorption window, narrow therapeutic index, and low soluble drugs. Um, and a few references, just therapeutic delivery. This is one paper that is for accordion technology and formulation approaches to enhance bioavailability of narrow absorption window drug. This is from Inventi Rapid Pharma Technology. Then the books which I referred, this controlled technology by Edit Wise. It is a Marshall Becker system, and pharmaceutical formulation edited by Mark Gibson, CRC Press, and also I have also referred Alton. So these are the sources of my information which I presented today. So I once again thank Dr. Ravi Kumar, Dr. Biswamohan, Dr. Niranjan, and other faculty members and non-teaching staff, and particularly the delegates who are attending this 2 weeks program, faculty development program. And as I told you, I am very happy to work in computers where hello, yes,
Yes,
yeah,
yes, sir. You are audible.
and thank you very much, Rai Kumar,
Yes, sir.
Mr. Mohan.
Thank you, sir. Nice presentation, sir. It will be very beneficial.
Whatever little I know, I have said with you.
Yes, sir.
Thank you very much. A few words, just I would like to. Delegates and my friends, really we are facing and passing through a very critical phase in our life.
Sir.
I think, therefore, we have to be very, careful, take care of ourselves.
Sir.
It's a bit of advice because day by day things are getting worse. The situation is has already assumed a very dangerous proportion, and therefore we have to confine as far as possible to our home and follow all the protocols as far as possible. But by God's grace, this very trying phase, and let us hope that soon we will overcome this pandemic and the normalcy will be restored. So once again, thank you and all the best to the delegates. Thank you very much.
Over to Rajaram, sir.
Thank you so much, Professor Bhanoji Rao sir, for sparing your valuable time to speak with our participants about gastrointestinal retentive drug delivery system. I am pretty sure that your presentation and discussion is for sure going to be much more helpful to most of our researchers who are on the same platform. May I now request our principal and coordinator of this FDP, Professor P.V.V. Ravikumar sir, to felicitate Professor M.E. Bhanoji Rao sir with an e-certificate of appreciation as a token of our affection and respect from entire RITS community.
Thank you, Professor Bhanoji, sir, my guru. for giving valuable information about gastroretentive drug delivery systems. It is always a pleasure and privilege to have you with us, sir. And thank you for all the support and guidance given to all the students and faculty members of Roland Institute of Pharmaceutical Sciences in the past, in general, and particularly to me. You're a big supporter of me. Because of your guidance and support only, I am here, whatever today I am. Thank you for that. And on behalf of the entire family of Roland Institute of Pharmaceutical Sciences, thank you for your contributions in the success stories and achievements of Roland Institute of Pharmaceutical Sciences. And as a token of love and respect, I would like to present this e-certificate of appreciation for your talk for today. Kindly accept it, sir.
Thank you very much for your affection and words, kind words. And I always cherish the moments which I have spent with all of you for a long time, and your association and the non-teaching staff, students, and management will be always well remembered. And
Some
yeah,
comments on chat box from your old students.
Bibu Prasad is asking, is telling,
Bibu Prasad.
you can see on the screen. Can you
Prasad
able to
Panda.
see, sir?
All are watching your presentation.
Yeah, he's Bibu Prasad Panda.
Yes, sir. You can see on the screen, sir.
Yeah. Let me see. Bibu Prasad. Yeah. Yes. How are you?
No, sir, not audible. He cannot speak, sir, here.
Okay. Thank you very much for your kind words. Kanucharan. Okay. Thank you very much. All the best.
हेलो सर. सर थोड़ा लिंक में ज्वाइन कीजिए.
Gautam Jana. Okay. He's the first batch student, I think. In B Farm. Okay. Thank you very much.
Sir, one more minute, sir. Bibu will be joining, sir.
Sir, actually we have to send the link, sir, na?
I have sent, sir.
Okay. Sir is having some network connection, maybe.
Suman, I think you can end the broadcast.
हाँ सर, आपण जॉइन करिवे. भानुजी दास सर अछंदी. Sir, just you announce for participants, then we can continue conversation. You just announce for participants.
Okay, sir. So, dear participants, with this, the scientific session 1 of Day 9 online FDP program has come to an end. We will be resuming the second scientific session at 4 p.m. today on the same platform. Till then, please stay safe and stay connected with us. Thank you, everyone, for
Okay.
your active participation and coordination.
Thank you. Thank you, sir.
Thank you. Thank you.
Sir, stay here. We can continue our
Sir.
conversation.
Okay.